Good afternoon, everyone. My name is Justin Meyer. I'm the Director of Development for the Department of Neurology. On behalf of myself and my colleague Stacy McAllister, thank you for joining us today for our first Brainwaves live event. Brainwaves is a monthly live virtual event featuring University of Pittsburgh and UPMC neurological experts who will present on a wide range of neurological conditions and disorders. Today, we're very excited to highlight Dr. Tim Greenamyre. Dr. Greenamyre is the University of Pittsburgh Love Family Professor, the Vice Chair of Neurology, the Director of the Pittsburgh Institute for Neurodegenerative Diseases, or the PIND, and also the Chief of Movement Disorders at UPMC. I could go on and on about Dr. Greenamyre's accomplishments throughout his career, but we only have an hour with you today. I want to highlight what many of you already know. He is one of the brightest scientific minds in Parkinson's research, an incredibly compassionate physician, and all-around great guy. Just a little housekeeping before we begin. All of our attendees' mics are muted, but you can type a question in the Q&A chat box, and we'll try to get to as many as we can in our allotted time. Dr. Greenamyre, we're so excited for you to be here with us today. So without further ado, please take it away.
Thanks, Justin and Stacy. This is a real pleasure to update everybody on the PIND, as we call it, the Pittsburgh Institute for Neurodegenerative Diseases. And I'm going to try something new. I'm going to try and give you a scientific seminar about what causes PD and how we might intervene therapeutically. Could I have the slides, Justin? Are they showing? I can't tell.
Good to go, Dr. Greenamyre.
Okay, thank you. So the PIND is located in what we call the Biomedical Science Tower 3, which is right across the street from the Kaufman building. The Kaufman building would be right over here. I hope you can see my little laser pointer. And we're located we occupy the entire 7th floor of this building here, which is modern open labs, as I'll describe. The concept of the PIND began in about 1997 when Bob Moore, who was a neurologist here at the time, and Michael Zygmunt, one of the pioneers of Parkinson's research, had an idea for the study of neurodegenerative diseases that would build on the research strengths here. And they really wanted translational research, which means research that translates from basic bench research into clinical practice. And it was important that they have close proximity of the labs and clinics so the scientists and physicians could work together, and that works out very nicely with the Kaufman building and our clinic right across the street. So I was recruited here in 2004 from Emory University to be the first director of the PIND. One of my patients dubbed me the PIND head after that. My mandate was to not only direct the PIND, but to create a clinical movement disorders division, the group that sees Parkinson's patients and related diseases. We've recruited a lot of faculty since then. We were designated an advanced center for Parkinson's research by the APDA in 2005. And we got to move into our brand new building in 2006. And some of you know Val Susky, one of our other movement disorders doctors. She is the director of the Center of Excellence for Huntington's disease, which we won't have time to talk about today. These are the diseases that the PIND focuses on. We'll focus today just on Parkinson's disease. And these are the things that we tend to look for. There are common features among the different neurodegenerative diseases, in particular between Parkinson's and Alzheimer's disease. And one of the themes across diseases is abnormal processing of brain proteins. The cellular garbage disposal systems don't work properly in these diseases, and abnormal proteins accumulate, and we'll talk about that a little bit. So what is the PIND? It is open labs. That means there's no walls between individual labs. One lab begins where another one ends on the next bench, and this results in a lot of crosstalk and collaboration between laboratory groups. It's scientists from a bunch of different departments, as I've listed here: neurology, pharmacology, neurobiology, and so on. There's 11 faculty members, 11 individual scientists who have lab groups, and about half of these are physicians, and several of those are listed as the best doctors in America or top doctors. So not only do we have physician scientists, but we have great physicians. There's PhD students and undergraduates and sometimes high school students working in the labs. We have people who are beyond their PhD and continuing to train. These are called postdoctoral fellows, and they come from all over the world. And it's a center without walls. So we include people from other departments who are located elsewhere as members of the PIND. This is a list of the current faculty and lab groups in the PIND. And as I said, about half of us are physicians who see patients in clinic. So at this point, what I want to do is give you a scientific seminar on Parkinson's disease. It's a little bit different. This is similar to what I would talk to other physicians or scientists about. And we'll see how I do in explaining it to you. So Parkinson's disease is a common disease. It affects about 1% of people over age 55, about 1 million people in North America. It comes in two basic flavors. One is genetic or familial cases of PD, and the much more common is sporadic, which is also called idiopathic, which means basically we don't know the cause. So about 85% of cases fall into this bin, the sporadic or idiopathic cases, and about 10 or 15% fall into the genetic or familial cases. We'll talk about the causes of Parkinson's disease. There's interest in environmental toxins, and I'll talk about that. There's interest in the cellular power plants in the cells called mitochondria and how they might not be functioning properly. And we'll talk about this culprit, alpha-synuclein, a protein that accumulates abnormally in PD and causes a lot of the problems. So first, a little anatomy of a nerve cell. Nerve cells contain a cell body, and the cell body is sort of the brains behind the nerve cell, and it has a long axon or nerve process that sends fibers and synapses and communicates with another cell down the line. Now, these long axons can be extremely long. You think about the nerve that controls your leg that goes from the cortex at the top of your brain all the way down your spinal column. And so that axon may be 4 or 5 feet long from a single cell. So here's a little anatomy at a more macro level. This is a slice through the human brain. We have the dopamine neurons, which have their home, their cell bodies are located here in a region called the substantia nigra, which means black substance, and I'll in a minute. And these nerve cells send their axons or their nerve processes up to a region called the striatum or the caudate and putamen up here. So this is the business end of the nerve cell. I'm going to show you in the next slide an actual picture of the human brainstem, the substantia nigra. And a few slides later, I'll show you a picture of the rat brain where we have pictures of the dopamine system up here at the business end and down here at the cell body end where the home of the cell bodies is. So the classical pathology, what goes wrong in the brain, is a loss of the dopamine neurons in the substantia nigra. And these are sections of the human brain. This is a normal human brain. And you can see this dark area here and here. This is the substantia nigra or dark substance. And if you looked with a microscope, there'd be hundreds of neurons in this slice and in this slice here. But they're largely absent here in a Parkinson's patient. Excuse me. In Parkinson's disease, many of the neurons that are surviving accumulate something called Lewy bodies, and I'll talk about that in this next slide. This is a picture of a neuron from somebody who has died with Parkinson's disease. This is the outline of the nerve cell. And you can see these big brown circular things in here. These are Lewy bodies that are packed into this nerve cell. They contain a whole bunch of different proteins, abnormal proteins. But the main protein that they contain is alpha-synuclein. And alpha-synuclein is an interesting protein. If you have a mutation in the gene that produces alpha-synuclein, you'll get a very aggressive form of PD. But if you simply make too much of the normal protein, you're at higher risk of developing the disease as well. So it's thought to be a key culprit, and it's part of the Lewy body. So there are a bunch of genetic causes of Parkinson's disease. And this is an out-of-date list, but there's more than 20 different known gene mutations that cause Parkinson's disease. And today we'll focus on two of them, this alpha-synuclein protein that I mentioned before, and a protein called LRRK2, which I'll talk about at the end of this. Alpha-synuclein, as I said, you don't have to have mutations in that gene. You can simply make too much of it, or you can have something that goes wrong with your cells so it doesn't clear it so well. So the garbage disposal system is defective, and you get too much synuclein. There are also environmental influences on your risk of developing PD. Things that increase your risk are pesticide exposure, rural living, agricultural occupation, and well water drinking. And these things are all probably related, and probably related through agricultural chemicals such as pesticides. Additionally, certain solvents, when you're exposed to them, increase your risk. And then there are things that decrease your risk. Tobacco smoking, coffee drinking, use of non-steroidal anti-inflammatory drugs like ibuprofen, and modest alcohol consumption. So the way I think about the cause of Parkinson's disease is you have a bell-shaped curve like this, and at one end of the spectrum you have the genetic causes of the disease that I mentioned before, but altogether these genetic causes of the disease account for only 10 or 15%. And at the other end you have even more rare single toxic exposures, sort of a massive toxin exposure that might cause Parkinson's disease. But for the vast majority of people, I think Parkinson's is caused by your total genetic makeup, things that increase or decrease your risk, and a lifetime of environmental exposures. And whether you're talking about genes that, when mutated, cause the disease, or toxic exposures, one thing that some of them have in common is that they impact the mitochondria, the cellular power plants in the cell, and cause power disruption and energy failure in the cells. So in this regard, I'm going to talk a little bit about a compound called rotenone. Rotenone inhibits the mitochondria. It decreases the energy that cells can produce. It also causes free radicals or oxidative damage to be caused. Interestingly, it's a natural product, and it comes from several different plant species. And because it was natural, it was assumed that it was safe. So even the organic gardening industry used to use it as an insecticide. So they didn't even have to disclose that they were using it. It's still used in lakes and reservoirs to kill nuisance fish or sample fish populations. So it's still in our water supply to some extent. And we decided to give it to rats to see what it would do, whether it would cause Parkinson's disease. And the answer is it did. So this is the rat version of the dopamine system. This is the business end of the nerve cells up here. And this is the substantia nigra, where the cell bodies are located. And this is a normal animal. This is an animal that's had a little bit of rotenone, more rotenone, and longer rotenone over here. And you can see that you lose the business end, the nerve terminals of these rats is degenerating. And eventually you get dying back and you start losing the cell bodies too, shown by the arrows and arrowheads. So this was really the first proof of concept that if your mitochondria, the cellular power plants, aren't working well, and in this case we inhibited them or poisoned them with a pesticide called rotenone, that can lead to degeneration that looks like Parkinson's disease. What made the model more interesting was the fact that for the first time in an animal model, we could produce something that looked very much like a Lewy body. So here's a dopamine neuron in a rotenone-treated rat that has those round inclusions that deform the cell that I showed you in a human brain. This is a rat brain. And this is higher power, showing the Lewy bodies in this dopamine neuron, and mini Lewy bodies starting to form in this other dopamine neuron here. Excuse me. So this was proof of concept then that mitochondrial poisoning could cause the pathology that looks like Lewy bodies in rats. Now, this gives me an opportunity to highlight the research of Dr. Sarah Berman. Some of you know is one of our Parkinson's doctors and listed as one of the best doctors in America. Sarah is known internationally for her work on the basic biology of mitochondria in neurons. But lately she's taken her interest in mitochondria and done more translational or clinical work. And she is now working in a project that's just beginning to use PET scanning to look at mitochondria in the living human brain. And these are some of the first images from a volunteer. In this case, this is a normal, healthy volunteer who got injected with this PET tracer and then got a PET scan. And you can see where the mitochondria are located in this rainbow scale, where the reds and yellows and oranges are more binding and the blues are less binding. So this is the first map of the mitochondria in the human brain that Sarah is developing. And she's going to look at how that changes in Parkinson's disease and in dementia with Lewy bodies. So it's a very exciting project. I mentioned that certain solvents are associated with an increased risk of Parkinson's disease. And one of these is trichloroethylene, or TCE. TCE is used commonly. It is a degreasing agent used to clean machine parts. It's really ubiquitous throughout the environment. It gets in through spills and just common use. It's in the groundwater, it's in the well water, and it's in certain EPA Superfund sites and places where there have been massive spills like Fort Lejeune or Camp Lejeune in North Carolina. The reason we're interested in TCE is that epidemiologic studies that have looked at people and what they get exposed to and what diseases they develop have identified TCE as causing a 6-fold increase in your risk of getting Parkinson's disease. So somebody in my laboratory began giving rats TCE at normal concentrations that people might get exposed to it, and found that it caused accumulation of this protein alpha-synuclein in dopamine neurons. So this is a normal rat here treated with placebo, and this is a dopamine neuron, and you see very little of this alpha-synuclein protein in that dopamine neuron. Here's an animal that was given TCE by mouth, and you can see that the synuclein is much more intense, and there's a particularly toxic form of synuclein accumulating as well. If synuclein is toxic, the question is, does it kill the dopamine neurons? And the answer is yes. So here's the substantia nigra where the cell bodies are located. And this is a placebo or vehicle-treated animal. And this is a TCE-treated animal. And you can see the marked loss of the dopamine neurons here. So this is reproducing what happens in Parkinson's disease. So I've talked a lot about alpha-synuclein and the reasons that it is causing cell death and degeneration in Parkinson's disease. And this gives me a chance to highlight work of my colleague Ed Burton, who's another Parkinson's physician, but an outstanding basic neuroscientist. And Ed is interested in, among other things, gene therapy approaches to Parkinson's disease. So I showed you this slide before. This is what happens when you give rotenone to an animal. And the loss of dopamine, or these dopamine lesions, as we call them, are bilateral. That means that they occur on both sides of the brain. And they're symmetrical, which means they're the same. They don't differ significantly from one side of the brain to the other. So they're bilateral and symmetrical. Here's another view of a rotenone-treated animal. And the colors have been switched around here, but here you can see the loss of the dopamine system at the business end of the neurons here, where the synapses are. And it's bilateral and it's symmetrical. The idea behind Ed's experiment in collaboration with our lab was to see if you use a gene therapy approach and knock down or reduce the amount of alpha-synuclein on one side of the brain. This is called gene silencing. So this side of the brain makes less synuclein. Can you turn this bilaterally symmetrical lesion into a unilateral lesion like this that is protected on one side with the gene therapy approach? And the answer is yes. So this is, again, the dopamine system where the nerve terminals are, and you can see the loss of the nerve terminals in a rotenone-treated animal. But on the other side of the brain that got the gene therapy, that side of the brain is completely protected. Down here is the substantia nigra, where the cell bodies are located. And you can see each of these little dots represents a cell. And you can see there's a lot less cells on the control side than on the gene therapy treated side. So again, it shows that knocking down or reducing the levels of alpha-synuclein can protect the brain. And this has led to a number of approaches to try and reduce the levels of synuclein or reduce its toxicity that are being tried now in clinical trials. Which leads me to talk about Lana Chahine, who directs our movement disorder fellowship for movement disorders doctors in training. Lana is an expert in alpha-synuclein in the clinical setting, and is the principal investigator, the senior person at UPMC and Pitt in a synuclein therapy trial. And the basic idea behind this trial is to give patients with Parkinson's disease antibodies administered through a spinal tap into the cerebral spinal fluid, and see if those antibodies can attach to the alpha-synuclein and clear it from the brain and improve Parkinson's disease. This is a large international trial. Each of these little green dots that I hope you can see is a different site in the US and across Europe that was participating in this trial. It's exciting because it was the first large trial to look at alpha-synuclein therapy. Unfortunately, the trial failed, and we could talk about the reasons for that, but I think antibodies aren't the best approach, and there are other trials coming down the pike that I think will be much more effective in this. But it shows you, I hope, how we came from looking at an animal model and the human pathology all the way to targeting alpha-synuclein in patients. In the last few minutes, I want to talk about another gene called LRRK2. LRRK2 is the most common cause of autosomal dominant PD. Autosomal dominant means that if you have one copy of the mutation, you will get the disease. And this is a very common mutation. It can account for 10% of genetic causes of cases of PD, and 3 or 4% of people with idiopathic or sporadic PD. This is the most common mutation called G2019S. And it varies by ethnicity. So people from North Africa, the gene or the mutation may be found in up to 30 or 40% of people. And in Ashkenazi Jews, it may be 13% of people. So it can be very common as a cause of PD. And as a result, pharmaceutical industry is working on developing what are called LRRK2 kinase inhibitors to block the toxic effects of LRRK2. Interestingly, LRRK2 has what's called incomplete penetrance. What this means is that if you have the mutation, you don't necessarily get the disease, but you're at much higher risk. The penetrance, the likelihood that you'll get the disease, goes up with age, but the reasons for the incomplete penetrance of this mutation are unclear and may mean that you need 2 hits to the system. You might need an environmental trigger along with having this mutation, for example. One of the questions that we were interested in was, does LRRK2 play a role in the more common sporadic cases? You know, if LRRK2 only is about 3 or 4% of total cases of Parkinson's disease, it's great if you can develop a therapy for it, but it'd be much better if you could develop a therapy for LRRK2 that would help people with idiopathic or sporadic PD as well. One of the fundamental problems with studying LRRK2 in idiopathic Parkinson's disease was that we didn't have a good way to tell whether it was active or not. And it's thought that its activity is what causes the disease. So if it's inactive, that's fine. If it's overactive, that's bad. So the question how to assess its function in Parkinson's disease. So for this, we developed a clever technique where we put a molecular beacon on the active form of LRRK2. So this is a cartoon of the LRRK2 protein. I know this doesn't mean much to anybody in the audience, but it's a very large protein. And we figured out a way to tag the active form of the protein with this bright fluorescent tag and determine whether it's active or not. And then we looked in the human brain. And what we're looking at here is dopamine neurons. These are the cell bodies in a normal brain of someone who has died and the brain of someone who's died with idiopathic Parkinson's disease. And these are the dopamine neurons. And what you can see here in red is that the bright red beacon is shining, indicating that LRRK2 is active in idiopathic Parkinson's disease, which was a major finding because nobody knew that that was the case. And it suggests that maybe if you can stop this activity, you can protect those neurons and improve Parkinson's disease. So what happens in the rotenone rat model? Can we see the same thing? So just pay attention to this left-hand panel here. This is a normal control or vehicle-treated animal. And this is the LRRK2, and you don't really see anything in these dopamine neurons. But in a rotenone-treated animal, you see the LRRK2 get turned on and become overactive. So that was exciting for us as well. And the question came up then, if rotenone causes alpha-synuclein to accumulate and causes the cells to die, as I've shown you, is it because LRRK2 is turned on and active? So we designed an experiment where we treated with rotenone, but we also inhibited LRRK2. We stopped its activity with a pharmacological drug called PF360. So let me walk you through this slide. This is the substantia nigra. Each dot represents a cell body of a dopamine neuron. This is a rotenone-treated rat, and you can see that there's many fewer of those bright white dots. The cells have died, and the animal is Parkinsonian. This is an animal that got rotenone plus PF360 to inhibit LRRK2, and you can see that by inhibiting LRRK2, we prevent the toxicity of rotenone. So this has some major implications. First of all, it suggests that LRRK2 plays an important, essential, central role in the vast majority of Parkinson's cases, and that rather than targeting only 2 or 3% of Parkinson's patients, those who have mutations, that LRRK2 inhibitors might be useful for individuals with idiopathic Parkinson's, even if they have no mutation. So that's great news for the pharmaceutical industry, because rather than a market share of 2 to 3% of Parkinson's patients, they may have a market share of 90 to 100% of Parkinson's patients. And it's also great news for Parkinson's patients with idiopathic disease, because they won't necessarily be left out of the therapeutics that affect LRRK2. So this is just another example of how we're trying to translate our research in the laboratories across the street from the Kaufman building into new therapies. So I've shown you a lot of research. I hope it wasn't too technical for you. We have challenges as we go forward in the PIND. We've been very successful in doing fundamental cutting-edge research, but we're facing challenges, and one of those is that most of our major equipment and instruments are 15 years old and obsolete or beyond repair. We have no operating budget or endowment, so we are applying for grants from the federal government and from foundations to do our research and fund our research. People often ask how expensive research is, and I would tell you that a typical medium-sized project costs about $1,000 a day to complete over several years. So it's an expensive endeavor. So we're thinking about ways forward. One of those is more partnerships with pharmaceutical companies and biotech, but those are often very targeted. You know, they're focused on what the pharmaceutical company is interested in, and where we can interact is good, but it's not more general than that. We're also concentrating more and more on philanthropy and hoping for philanthropic gifts. You know, every little bit contributes to the success of our projects. I don't want to quit before I tell you about active clinical studies for those of you interested in participating. The contact person for any of the research projects is Sherry Masovsky. This is her email, and I will leave that up while I show you the studies. For everyone, regardless of your disease or whether you're a Parkinson's patient or a spouse or caregiver, we have a research registry, and I would encourage everybody to sign up for this. It's very easy, and this gets you into our list of people who are interested in research so that we can call you when the opportunity arises for a new study. For Parkinson's patients who are just newly diagnosed and are so far untreated, we have an exercise trial to learn about the effects of high-intensity aerobic exercise on Parkinson's disease. And those of you who are my patients or other neurologist patients have probably heard repeatedly that exercise is good for your Parkinson's disease. Then a variety of studies are available for people with Parkinson's disease at all stages. One of these is in light PD, to learn if light therapy reduces sleep problems and improves quality of life. A relatively new one is the Topaz study to learn if an osteoporosis medication called zoledronic acid, or Reclast, can prevent fractures in people with PD. And then I already showed you Dr. Berman's PET scan study. And there's another study for those people who have what's called orthostatic hypotension, which means when you stand up, you get dizzy or lightheaded or pass out. This is a drug to help with that. And then for people with dementia with Lewy bodies, Dr. Berman has the DLB consortium study, which is pretty intensive, but important. It includes an annual spinal tap and blood collections, along with cognitive testing. And that's all for today's presentation. I hope it was somewhat comprehensible, at least. And with that, I guess we can go to the question and answer with Justin.
Thank you so much, Dr. Greenamyre. What an incredible and insightful presentation. We're so fortunate to have you with us at the University of Pittsburgh and UPMC. Just before we begin the Q&A portion of our event, I'd just like to take a moment and thank someone who's very special to us at the PIND. Today just felt fitting since I know he's on the call with us. His name is Sean Logan. Sean came to us a number of years back and said he wanted to do something to help support research in Parkinson's disease. What we didn't know that day is the incredible impact that it would make for seemingly all of us. Through his work and fundraising with his creation of the PIN 5K, the numerous connections that he's made for us, in the awareness that he's brought to the PIN is just simply immeasurable at this point. Sean has raised over $1.3 million to support the work of Dr. Greenamyre that he just presented. So Sean, you, your wonderful family and friends are absolutely amazing human beings, and words cannot explain how grateful we are for you. So thank you. We're going to now begin the Q&A portion of our presentation. We'll try to answer as many questions as we can in our allotted time. We do have a lot of questions, so we'll try to get to them. Dr. Greenamyre, just sort of right at the end there with your call to enroll in research studies, do you need to live in Pittsburgh to enroll in research registry?
No, you don't have to live in Pittsburgh for the research registry. Where you live may be important for certain clinical trials. You know, if they require frequent visits and that kind of thing, just logistically and in terms of convenience and transportation and things like that. But to enroll in the research registry and hear about the trials, absolutely not. We have people from all across the country.
Okay, great, thank you. How much TCE exposure is represented in the experiment you highlighted?
It was at the upper end of what are called NIOSH daily limits. The NIOSH, which is National Institute of Occupational Safety and Health, comes up with chemical limits based on certain toxicities, and this is sort of at the upper range of what's permissible on a daily basis for humans. So it's a realistic exposure, I think.
Okay, thank you. Timely question here. How is COVID-19 affecting Parkinson's patients?
That's just becoming a little bit clearer. It looks like Parkinson's patients may be hit harder with the disease, although it's still not considered a pre-existing condition that will get you earlier vaccination priority. It also looks like potentially Parkinson's patients may be more likely to get COVID-19. So you're more likely to get it and it's more likely to be severe. So I strongly encourage everybody to maintain social distancing, wear your mask, and get vaccinated as soon as you can. While I'm on the topic, a number of patients understandably write to me and say, can you get me an earlier vaccination? I cannot. I've written a couple of letters for people to say that they have Parkinson's disease. Please consider giving them a vaccine. I don't know how that's met, what success that has caused or not. But I don't have any way to prioritize people. I can't even prioritize it for my wife.
Thank you, Dr. Greenamyre. Along the same lines, do you think that COVID-19 could trigger Parkinson's disease?
I think there's a possibility that it could, in the sense that young people who get COVID-19 may be predisposed years later to get Parkinson's disease. And I think we won't know that for many years, like the Spanish flu did at the turn of the 20th century. It may be that people who get exposed have a predisposition years later. The other way that it might seem to cause Parkinson's disease is that is a phenomenon many of you have probably noticed, and that's when you get sick or you have major surgery or you have a major life event. That seems to be when your Parkinson's symptoms first appeared. And it isn't that those events or the sickness caused the disease, but they just brought out the symptoms a bit earlier than they would have come otherwise. Hopefully that makes sense to you.
Thank you, Dr. Greenamyre. Is it true that Parkinson's disease is the fastest growing neurological condition?
It is. It's the fastest growing neurodegenerative disease, yes. And there is a growing thought that has to do with the environment and environmental toxins, as I was discussing.
I guess a nice segue into the next question. Have you identified any environmental triggers? Is there anything new that you are concerned about?
I think there's reason to be concerned about a number of pesticides, aside from rotenone, which is an insecticide. There's paraquat, which is used in the farming industry to get rid of weeds before planting crops. So there's good evidence that paraquat may cause Parkinson's disease as well. I think there's a lot of things that we get exposed to that we simply have no idea about whether they do this kind of thing. The testing of toxicities of chemicals in the environment is really pretty minimal. I mean, we assume that things are tested and safe, but that's really not often the case. And the kind of testing that you have to do to figure out whether it has neurotoxicity and causes something like Parkinson's disease is long and expensive and not required. So I think there's a lot more to come.
Okay, thank you. Is PF360 being used regularly now? What are the side effects and how is it administered?
PF360 is really a tool compound. It was made by Pfizer, and unfortunately Pfizer's gotten out of the nervous system business. They had a program in Parkinson's disease and no longer are. So it's what's called a LRRK2 kinase inhibitor, and it was more proof of concept that this could help. The companies that I think are the farthest along in looking at these kinds of drugs are a collaborative effort between a company called Denali and Biogen, and they are looking at LRRK2 kinase inhibitors and have done some safety trials so far, but don't have any evidence yet in terms of Parkinson's patients. So far, they seem to be pretty free of severe side effects. What I will say, because people are always interested, is the first clinical trials, when these begin more widespread clinical trials in hopefully the idiopathic or sporadic cases, the first trials will be targeted to those with de novo disease, those patients who are newly diagnosed and untreated. So people who are already on medications or have been diagnosed for a few years won't be eligible for the first clinical trials. Hopefully when it passes the initial trials with the very early patients, then it will become available for people who have had the disease a bit longer.
Thank you, Dr. Greenamyre. We have some comments. I'm just going to portray those across to you here. Dr. Greenamyre, thank you for your dedication in the clinic and in the lab. Thank you for presenting to us today. You are a wonderful and compassionate physician. I'm proud to support this work.
Make me blush.
Yeah. Sean, congratulations on your fundraising achievements for the PIND. Your 5K is a special event that I always look forward to each year. So some nice comments there.
Yeah, I have to second that comment about Sean. Sean is a wonderful human being and has done so much for the PIND and for Parkinson's disease. He has my eternal gratitude.
Thank you, Dr. Greenamyre. I know there's a lot of questions about people's daily jobs and how that might affect their disease. Here's one: could daily use of a gas-powered leaf blower impact my Parkinson's disease?
Not as far as I know, no.
Okay. Are the alternative treatments for PD, example, deep muscle massage, CBD, acupuncture, how do you feel about those?
I feel that there's no standard of scientific proof that they work, but many people swear by them. You know, if they feel good and they relax you and help your rigidity or your tremor, or just help your anxiety, which is a common problem, I'm all for it as long as I think they're safe. So people often in my clinic bring me a list of the supplements that they're on, and as long as I think they're safe, I have no problem with them continuing, as long as they also don't interact with their medications. The parallel question that often comes up is, what about medical marijuana? And again, I would say, if it helps, that's great, but there's no good systematic proof of it. It's been impossible to do scientific medical trials with it because it's been prohibited by the federal government until about now. It's starting to change. One of the problems with medical marijuana is that there's very little standardization. So what you get at one outlet may be completely different from what you get at another outlet, and you're not comparing apples to apples and oranges to oranges necessarily. So it's still a little bit of the Wild West out there, but I have no problem with it if people think it helps.
Okay, thank you. We have a question here. How do we find out about the philanthropic opportunities? Who do we contact? I'll take that one. I know a lot of you have engaged with myself or Stacy McAllister. Please feel free to reach out to us, and we can help you with any of your thoughts or any way that you want to support Dr. Greenamyre's work. It is vitally important to the research that he does. Dr. Greenamyre, how can we get more people enrolled in trials? Won't that help us find a treatment or cure faster?
Yes, trials are the key to proving that something does or does not work. And I would say the Michael J. Fox Foundation has made major efforts to try and encourage people to enroll in trials, which I think is great. There's something called the MJFF Trial Finder, where you can plug in where you live, what your symptoms are, when you were diagnosed, and that kind of thing, and see what trials around the country that you might be eligible for, which are in your region, and so on. But I entirely agree that participation in clinical trials is extremely important.
Thank you. How is it determined if one has genetic or idiopathic PD?
That's a great question. The first real hint that somebody has genetic PD is from their family history. So that's why we all often ask a new patient, who in your family has had PD or something like PD? And if there's somebody, then we often take a more detailed family history. You know, if it's an aunt, we ask about the aunt's brothers and sisters. and their children, you know, we will dig into it. That's the first clue. In some cases, it's clear that Parkinson's is running right through a family, and that's a major clue. We don't use, for clinical purposes, we don't use genetic testing very often, and there's a couple of reasons for that. One is that the genetic test, if you're negative, you don't have a mutation, it doesn't mean you won't get Parkinson's disease. So this is for pre-symptomatic testing. If you don't have one of the Parkinson's causing mutations, it doesn't mean you won't get Parkinson's disease. And the other problem is exemplified by what I said about LRRK2 earlier, that it doesn't have complete penetrance. So in many of the mutations that we can detect with genetic testing, if you have the mutation, it doesn't mean you will get the disease either. And thirdly, you know, even if you have a genetic form of Parkinson's disease, it doesn't change how we will manage the disease at this time. Now that may change. There may be progress such that if you have a certain kind of mutation, we can use a certain kind of drug to treat that type of Parkinson's disease specifically. That's what's called personalized medicine. We're not there yet, but that's the goal. So right now, genetic testing just isn't helpful too much clinically, but for research purposes, depending on where you get your care, we will do genetic testing.
Thank you, Dr. Greenamyre. Dr. Greenamyre, do you think there is a link between sleep interruption, sleep apnea, and PD?
There's not a clear link, although many patients with Parkinson's, and I guess it depends on how the question was intended. Do I think that one causes the other? I think that there's no evidence that a sleep disruption or sleep apnea causes Parkinson's disease. Many patients with Parkinson's disease do have sleep abnormalities. The most common and best recognized is REM behavior disorder, which is where you act out your dreams and fight and thrash out, kick or hit. This is a question we often ask about in the Parkinson's clinic. That particular symptom is probably one of the earliest in many cases of Parkinson's patients. So one of the interesting things is we recognize Parkinson's disease as tremor and rigidity and gait and balance problems and so on. We think about the motor signs of Parkinson's. But Parkinson's disease often begins with these sleep issues in the REM behavior disorder, or with constipation or a loss of sense of smell that people have had for years before they develop things that are finally recognized as Parkinson's disease. But it's actually the beginnings of Parkinson's disease in many cases.
Thank you, Dr. Greenamyre. There's been a couple questions about whether the presentation will be available afterwards, and yes, it will be. We're recording, and please feel free to reach out to myself or Stacy, and we can get your recording after it's done. And please feel free to pass it along to those that you think may be interested in it. When do you think precision medicine will be available for PD? It seems to be common in many other diseases.
That's a great question, and it really depends on our getting a better handle on what is causing Parkinson's disease and how we can arrest it. So the best example I can give you is for LRRK2, where if you have a LRRK2 mutation, these kinase inhibitors that are being developed may be great for you, although I think they'll be great for everyone with Parkinson's disease. But the general idea is that if you have a mutation in a particular gene, and we have a way of attacking or correcting that gene, then we'll have personalized medicine. And I think we're probably 5 to 10 years away from having that more widely than we do now.
Thank you. The SPARKS trial was infusion of antibodies into the bloodstream. Since it failed, is there a better approach?
Yeah, good question. The general idea is to try and decrease the amount of alpha-synuclein or reduce its toxicity, and both of those approaches are being used. So there is a gene therapy approach called antisense oligonucleotides, which means that you're treated with these things called ASOs, antisense oligonucleotides, that get into the brain cells and cause those brain cells to produce less alpha-synuclein protein. I think that's a very promising approach, and that approach is also being used for LRRK2. So I think there are novel gene therapy approaches that can be applied to reducing alpha-synuclein.
Okay, thank you. What kind of new technology equipment would have the biggest positive impact for the Institute right now?
Right now, it would probably be what's called a laser scanning confocal microscope. Ours is 15 or 16 years old at this point, and it's beyond repair. It's sort of limping along right now. Unfortunately, they cost probably about $400,000, and the Institute has two that are dying, along with another major piece of equipment. So we're talking about, just to replace equipment to make us whole, we're talking about $1 million or so, and that's not easy to come by.
Okay, thank you, Dr. Greenamyre. Looks like we have time for maybe one more question here. Dr. Greenamyre, what makes you excited about the future for Parkinson's research?
That's great. That's a great question. Patients who come to see me have heard this more likely than not, but I say there's no good time to get a diagnosis of Parkinson's disease, but this is the best time in history to get a Parkinson's diagnosis, because I think we really are on the cusp of what are called disease-modifying therapies. You know, our treatments right now are symptomatic. We can take care of the tremor or the rigidity or that sort of thing, but we don't change the overall progression of the disease. I think we're getting there with some of the approaches that I tried to inform you about today, and I think I'm really excited that we're going to have an impact on the course of the disease in a big way. And just to give you an idea of how this might come to pass, you know, if we have something that slows the progression by 50%, so it still progresses, but at 50% slower, you can imagine that given the age of diagnosis for most people, 50% slowing would mean they'd have a great quality of life until their normal, natural lifespan ended. So I think we can have realistic goals and get there in the reasonable future. So I'm really excited that we're going to change the course of PD.
I'm glad that was our last question, Dr. Greenamyre. Nice way to end, a nice positive note. Again, thank you so much. What an incredible presentation. I hope everyone enjoyed it. I'm sure they did. If you'd like a copy of the presentation, please feel free to reach out to myself or Stacy, and we'll make sure you get that. Again, thank you for joining us today, everyone, and Dr. Greenamyre. Everyone have a great day. Stay safe. Be well.
Thank you, everybody. Goodbye.
Bye. Thank you.